Yes. First one, peroxisomes. Can you explain the paper as previous? Previously, you have done a great job as I remember. If you can explain the paper because here you only. Yes. First one, peroxisomes.
Peroxisome
Oh,
only found in plants. False.
why? Even in animals, we can find peroxisomes. Can you mention the microbody type which can be seen only in plants?
Glyoxysome.
Yeah, only glyoxysomes, ne? Glyoxysomes. Then we can make a small note here.
Teacher, mic is muted.
After finishing, let me know. There is a sound disturb. That is why I muted it.
Teacher, then lysosomes are also present in both animals and plants.
Lysosomes, yes. Lysosomes are made up of Golgi complex, ne? Yes. Plants also have to if there is a disturbance, let me know.
Okay, teacher.
Okay, if it is hard for you, let me know. I will stop then. Yes, lysosomes also can be found in plants because they are actually self. Actually, they are suiciding organelles. Ne? Therefore,
Yes,
plants
teacher.
also, yeah, plant cells are also aging. Then they have to cell repair in their bodies, and organelles also have to repair. Once they have broken or destroyed, they have to break down into their simple molecules. Therefore, lysosomes also can be found in both animals and plants. Then second one is contributing C3 photosynthesis. Then
Correct.
correct, very good, correct. As I mentioned in your theory note, which kind of organelles are contributing in the photo respiration in C3 plants? Which kind of organelles? What are the two organelles, including what? Peroxisomes. First one is peroxisomes. Second one is actually the peroxisome. Other one is what? Photorespiration in plants. C3 plants.
Mitochondria.
Very good. First one is mitochondria. Second one is peroxisome.
Okay, teacher.
Two membranous
Chrome,
organelle.
wrong,
Yeah.
single membrane.
Yeah. Then
Contains reducing enzymes. False.
then they should.
It contain oxidizing
Yeah,
enzymes.
yeah, yeah. It should oxidize in enzymes. Use a red point for writing these things actually. Then you can easily catch the point in your exam
Contain a
days.
crystalline matrix. False.
Crystalline matrix actually include. but in here it is not mentioned, therefore we have to ignore this one. Actually the matrix of the lysosome is a crystalline thing. If you want you can remain it, but it is not included in the new syllabus as a crystalline matrix. In your resource book it is not included. Yes, then answer is number 2. Answer is number 2. Second one.
Incorrect statement. Tissue is a correct cell collection which is structurally accumulated to perform a specific function but not functionally accumulated. Wrong, because it is functionally also accumulated.
Yeah, have you finished this one?
Yes,
Have you
teacher.
noted? Yeah, very good. Second one, incorrect. First one incorrect. In second question, both structurally and functionally accumulated. Second one.
Stopping of the biological activities are called as the cell death, correct? Third one, cell aging is the replacement of the maximum activity of cell, correct?
Correct. As an organism, eh? As an whole
Neurons,
organism. Yeah.
neurons are differentiated to transmit impulses, correct?
Correct. That means they have an inability to divide further. Okay, once they are differentiated, once they have differentiated of any kind of cell, animal or plant or any kind of cells, once it has differentiated means differentiating for a specific function. Okay, differentiation for a specific functions. Once it is differentiated, it cannot be divided further. Are we unable to divide further, unable to produce new daughter cells. Once it is differentiated means it is differentiated to a particular function. What about the particular function of neurons? That means differentiated cell. What about the differentiated function in neurons?
Transmit impulses.
Yeah, impulses transmission. Very good. If you have anything to write, note down things, stop me and you can continue it. Then we can stop it and you can do it. Then we can continue.
Okay, teacher. Organizational levels of organisms are arranged from simple level to complex level as atom, molecule, organelle, cell, tissue, organ system, organism, population, community, ecosystem, biosphere, correct?
Correct. Yes. What are the two key points of the what? How we can identify the living characteristic regarding this? One of 7 living characteristics.
All organisms are made up of one or more cells.
No. Living characteristics. Living characteristics of organisms. How we can say this is an organism or not? How we can say you or me or any kind of organism is an organism or not? How viruses are differ from organisms?
They should show the 8 characteristics of organisms at the same time.
Yeah, 7 or 8. 7 or 8.
8.
First one. Order 7 neither. Order and organization, metabolism, growth and development, irritability and coordination, adaptation, reproduction and heredity and evolution. Other than that,
7 only.
7.
Station.
All the 7 characteristics should be presented in that particular organism. That is how we can say it is an organism. So can you mention the living characteristic regarding the 5th statement of question number 2?
Order and organization.
Yes, order and organization. What are the key points? What are the two key points of order and organization? Uh, how we can create this hierarchical organization? First one is the organism and the structural and functional unit of the organism. What?
Cell.
Cell. Yes. Cells produce what?
Tissues.
Yes. Cells. Cells and cell and organism is the two key point. Cell and tissue. Then
Organ.
yeah, organ.
Organ system. Organism.
Organism. In an ecological system, organism can be identified as another term.
Individual.
Organism or individual. Then how cell is produced?
A combination of organelle and organelles are formed by molecules. Molecules are formed from atoms.
Yeah, which kind of molecules? Simple and then complex. Neither. Organelles, complex molecules, simple molecules, then atoms. Then organism or individual, then
Population, community, ecosystem, biosphere.
biosphere. Very good. Biosphere has three components. Can you mention?
Lithosphere, atmosphere,
Mm.
hydrosphere.
Lithosphere, atmosphere, and hydrosphere. If you have anything to write a note, you can note. Otherwise, we can continue.
Okay, teacher. Completed, teacher.
Third question. Third question.
Does not contain in the cytoskeleton microtubules, microfilaments, actin filaments, myosin filaments, intermediate filaments. Answer is myosin filaments.
Answer is very good. Answer is myosin filaments. How many components are in the cytoskeleton?
3.
Three components. First one is
Microtubules,
yes, microtubules,
microfilaments or actin filaments,
microtubules, then microfilaments.
filament,
Yes. Can you remember the monomer of microtubules?
tubulin,
Very good. Tubulin protein. And microfilament?
actin,
Yeah. Both the proteins or carbohydrates or lipids or
proteins,
yeah. Third one is
intermediate filaments.
intermediate filaments. Very good. Answer is number 4.
Okay, teacher.
Yes. Next one.
Incorrect about the cytoskeleton. Contain only in the eukaryotic cell. Wrong. It is a dynamic structure, correct. Involved in the cell shape, correct. Mainly it is composed of lipids and fibers, wrong. It is distributed through the cell, wrong.
Answer is
Fourth
so
one,
fourth one. Actually, containing only in eukaryotic cells, as previously we have discussed, cytoskeleton, normally we say cytoskeleton can be presented in eukaryotic cells only. Ne? Can you remember?
next is
Yeah, therefore we can take it as a correct answer. Correct. Second one, dynamic structure, correct. Third one, involved in the cell shape, especially in where?
animal
Involved
cell.
in animal cell. Because of the absence of
Cell wall.
cell wall. Mainly it is composed of lipids and fibers. Why it is
False.
incorrect?
This consists of proteins and fibers.
Which kind of fibers?
Protein fibers.
Yeah, therefore mainly it is composed of mainly proteins. Protein fibers. Protein fibers means again proteins.
Yes.
Fifth one, it is distributed throughout the cell. Yes. Answer is number 4.
Okay, teacher.
Then
Not a function of smooth endoplasmic reticulum. Storing calcium 2 plus ions, correct.
correct.
Producing transport vesicles for transporting materials within the cell, correct. Detoxification, correct. Metabolism of carbohydrates, correct. Synthesizing glycolipids, wrong.
Synthesizing glycolipids means a function of
Endoplasmic reticulum.
which endoplasmic reticulum?
Rough endoplasmic reticulum.
Yeah, rough endoplasmic reticulum. It is a function of last one, fifth one.
Yes, teacher.
Yes, fifth one is the answer. It is a function of RER. Then mention common function or functions of SER and RER.
Producing transport vesicles.
Here we are talking about SCR. Therefore, mention mention the unique functions for SCR.
Storing calcium 2 plus ions.
Storing calcium 2 plus ions.
Metabolism of carbohydrates.
Yes, other one is storing calcium 2 plus ions and
Detoxification.
yeah, detoxification. Detoxification enough. Then metabolism of
Carbohydrates.
which kind of lipids can be synthesized by SCR?
Oils, steroids, phospholipids.
Yes. Oil, steroid, phospholipids. SCR. Which kind of lipids can be synthesized by RER?
Phospholipids.
Yeah. That means phospholipids can be synthesized by both SCR and RER.
Teacher, but RER use phospholipids protein molecule and makes its own membrane.
Yes, but as a single function, we can mention a synthesizing of membrane phospholipids.
Yes, teacher.
Yes. Can be synthesized by both SER and RER. Phospholipids, and other one is any other lipids. We can mention glycolipids also. Here they have mentioned phospholipids, proteins and carbohydrates are accumulated, connected with each other. Then after connecting phospholipids with uh, carbohydrates, which kind of structure produced?
Glycolipids.
Yeah, here mentioned glycolipids also. Glycolipids, the lipid part is also a phospholipid, a kind of phospholipid, because carbohydrate chains are attached to the phospholipid bilayer. Other than that, glycolipids and other thing is what? Glyco?
Protein.
Yeah, glycoproteins.
Okay, teacher.
Yes, next one, 6.
Actively involved in the cellular respiration, mitochondria.
Mitochondria, very good. Now, even we haven't uh, do the cellular respiration part. Up to now, I have mentioned what are the 3 steps of cellular respiration. 3 steps. Why we say mitochondria is actively involved in cellular respiration, as it has 3 steps. First step is what?
Glycolysis.
Very good, glycolysis. Second one is
Krebs cycle.
Krebs cycle. Third one is
Electron transport chain and oxidative phosphorylation.
chain and oxidative phosphorylation. Hari, mention what are the relevant places in the cell in each step. Glycolysis.
Glycolysis in cytoplasm.
Very good. Cytoplasm. Krebs cycle.
Mitochondria.
Mitochondria. Where?
Matrix.
Yeah. Matrix. And third one?
Cristae of mitochondria.
Yes, that means the inner. That means the cristae means the inner.
Folding.
Inner.
Folding.
Cristae is a part of inner
Membrane.
membrane. Normally we say it is the inner membrane. To increase the surface area, inner membrane has produced cristae. Therefore, mentioned as the inner membrane. Okay, inner membrane. Cristae is an adaptation for it of mitochondria.
Okay, teacher.
Why didn't you? Why can't you switch on the camera at this moment? If you can't, oh, it's okay. Otherwise, you can switch on your camera as previous. Okay. Then 7.
Correct about plasmodesma. When we observe
Plasmodesmata. Yes.
by naked eye, wrong.
Wrong. Yes, I will explain because I haven't covered this up to now. Plasmodesmata is a type of cell junction. It can be only found in plant cells. As animals, we have some junctions in between our cells. Gap junctions, anchor junctions, like junctions. At the end of this cellular basis part, we will discuss. Anyway, in plant cells, we can see these structures we called as plasmodesmata. This is the cell wall, primary cell wall. Plasmodesmata can be seen in their primary growth. Not like us. Plants have two types of growth we called as primary growth and the secondary growth. In the secondary growth, the secondary cell wall is deposited. In their primary growth, only the primary cell wall is presented. On the primary cell wall is presented. This is the primary cell wall. This is the primary cell wall. Okay. And this is the central vacuole, nucleus, and everything. No, you know these things. There are some. This is one cell, and those are adjacent cells. Those are adjacent cells. There are some pathways which are called as plasmodesmata. That is how materials are transported among the cells in the tissue. There are some pathways. There are some pathways. This is the cell wall and this is the plasma membrane. Plasma membranes are interconnected via this plasmodesmata. This particular junction pathway we called as plasmodesmata. These openings we called as pits. This is called as the plasmodesmata. It is the cell junction type of plant cells. Can be observed by the naked eye, as you have mentioned, wrong, going through the cell walls of plants. Correct. They are non-living.
Wrong.
Wrong. They are actually living. After depositing the secondary cell wall, it becomes the dead state. Even they are dead state or non-living state, pits are remaining, but plasmodesmata cannot be seen. Plasmodesmata is a living structure. Actually, it is a cytoplasmic projection because cytoplasm of this cell is connected with the cytoplasm of this cell. Inside the plasmodesmata, cytoplasmic components are included. Therefore, plasmodesmata can be considered as a cytoplasmic projection-like thing. That is why. Yeah. Not essential in living characteristics here.
Wrong.
Wrong. Found in some animals, only some animal cells, not all, only some.
Wrong.
Wrong. Never. Only found in plants. only found in plants. Next one, you can explain.
Correct about centrioles. Microtubules are involved in the formation. Correct.
Microtubules or microfilaments?
Filaments.
Microtubules or microfilaments?
Tubule.
Tubules. Tubules. Microtubules. Which has 13th microtubules. The 13th microtubules.
Yes, teacher.
13 microtubules.
Microfilament.
13 microtubules.
Tubulin.
Tubulin. Tubulin. 1 microtubule. Microtubule makes not only centrioles, other than that, centrioles, other than that,
Asterspiral.
other than that. Initially, we have discussed before centrioles.
Flagella.
Yeah, flagella.
Yeah.
Cilia. Yeah, then microtubules are involved in the formation. Microtubules or microfilaments.
Microtubules.
Yeah, microtubules. Produce aster spindle in the cellular development, correct? Centrosome and
Teacher.
centriole are
Not cellular development now during cell division.
in the cell division. Very good. Centrosomes and centrioles are same in their structure.
No.
Okay, keep it 1 minute. Arranged in the cell close to the nucleus.
Correct.
Yes, you can show it. How we can see. Yes, this is the nucleus. The centriole can be seen like this in both animals and plant cells. Animals and plant cells.
Not in plant cells.
Yeah, only in animal cells. The whole structure we called as what?
Centrosome.
Centrosome. Those are centrioles. Difference between even they have these three are made up of centrioles and flagella are made up of microtubules. The difference?
Flagella and cilia have a 9+2 arrangement, 9 doublets.
Doublets, yeah.
Yes, cell body only have 9+0 arrangement. In centriole, it have a 9+0 arrangement, but it's triplet, 9 triplets.
Yeah. Here, even in the basal body, it has 9+0. Here also 9+0. The difference is here microtubule doublets are included. Here microtubule triplets are included. Then
Not an organelle, correct?
correct. Then answer is number 5. Arranged in the close to nucleus also correct. Same in their structure. Structure means the composition like thing neither. Structure. Same in their structure. No, we can't say it because in centriole, the structure of centriole, only one set of 9+0 microtubule arrangement. In the centrosome, 2 centrioles, that means 2 sets of 9+0 microtubule arrangement. Therefore, structure different.
Yes.
Produce astaine spindle in the cellular development, correct? Yes. Reducing sugar. Again, can you mention the
Yes.
reduce?
Then 8th one answer is first. Answer 4th and 5th.
Yeah. Cellular development, cellular division.
Okay, teacher.
Finished?
Yes, teacher.
Yes. Reducing sugars are what?
All monosaccharides and all disaccharides except sucrose.
All polysaccharides are not reducing sugars. Am I correct?
Yes.
All polysaccharides are non-reducing sugars. Am I correct?
Wrong.
Why?
Because polysaccharides are not considered as sugars, so they are
Yeah.
not either reducing or non-reducing sugar.
Yeah. Very good. Reducing sugar here is sucrose. Sucrose. Can you mention the relevant lab? I have asked. Okay. What is?
Teacher. Here, reducing sugar, sucrose is a non-reducing sugar. So the question should change, not non-reducing sugar.
Yeah, you can change the question according to your way. That means you can ask about the non-reducing sugar or as reducing sugars. 1, 2, 3, and 4 can be taken. Reducing sugars are all monosaccharides.
Okay, teacher.
Then
Correct about plasma membrane. It is mainly composed of carbohydrates, phospholipids, and protein. Wrong. Because mainly it is composed of phospholipids and proteins, then carbohydrates.
Yeah.
Phospholipid molecules can move and give the membrane a fluid character, correct?
Correct.
Peripheral proteins are tightly bound to the outer surface of the membrane. Wrong. Because they are loosely bounded.
No. It should not outer membrane. It should the inner membrane of the cell.
Teacher, peripheral membrane are also loosely bounded.
Loosely bounded. One thing is loosely bound. The other difference is
in the inner surface.
Inner surface. That means Have it mentioned loosely bounded like thing? Are there?
Yes, teacher.
Yeah. Loosely bound to the surface of the membrane. As this is a cell, this is the nucleus. When we cut one section of the plasma membrane, we can identify in this part. This is the inner surface, because this is the inner surface and this is the outer surface. In our previous diagrams we have changed inner and outer surfaces, but in here as we cut this one, we can draw our normal diagram, outer and inner, like that, outer and inner. Then peripheral proteins are loosely attached to the inner surface. Here inner surface is there. Here the inner surface is there. Therefore peripheral proteins are located here. That means here peripheral proteins are located in here, inner surface. The one is it should loosely bound.
Fourth one, the phospholipid bilayer helps adjacent cell communicate with each other. No. Because proteins only communicate with other cells.
Communicate. And what about the yes, proteins, embedded proteins, ne? Next one.
Hydrophobic tails of phospholipids bind to cell membrane fibers and help maintain cell shape. Wrong, because hydrophobic tails are inward to the hydrophobic head.
Actually, it should bound to hydrophobic head of the plasma membrane by phospholipid bilayer, ne?
Yes, teacher. Then answer is second.
Yeah. Third one. The next one.
3 genera with circular histones bound to DNA and several forms of RNA polymers respectively.
Circular chromosomes means what? Circular chromosomes. Means what about the domain? Circular chromosome having domain?
Prokaryotes.
Prokaryotes. Normally we can't say circular DNA of prokaryotes as chromosomes, but I have mentioned in molecular biology, normally we use the term chromosome to represent the circular DNA, but they are not true chromosomes. In here, circular chromosomes we can consider it as prokaryotic nuclear material DNA, cyclic DNA. That means it should domain archaea or domain bacteria. First answers, look at first answers. Thermococcus. Thermococcus is what? Thermococcus is what? Bacteria or Archaebacteria?
Bacteria.
Thermococcus.
Archaebacteria.
Thermococcus, how we can identify it is an Archaebacteria? Thermo means high temperate,
Heat.
high heat. Then it is an Archaebacteria. Hari? They are in extreme environments. Ne? Thermococcus in an Archaebacteria.
Yes.
Then Methanococcus. Methanococcus.
Bacteria.
No. High methane concentrated places living Archaebacteria. Look at your chart. That means no examples here. Neither. In your unit 3, actual examples are included in unit 3. Okay. The next one is Anabina. Anabina, nostoc are cyanobacteria. Cyanobacteria is an archaebacteria or normal bacteria. Cyanobacteria is an archaebacteria or normal bacteria. Cyanobacteria, Anabina nostoc-like cyanobacteria is an archaebacteria or normal bacteria. Can't you hear me? You can't hear me? Can you hear me now? Thermococcus. Thermococcus is an archaebacteria. Methanococcus is an archaebacteria. Thermococcus is an archaebacteria. Methanococcus is an archaebacteria. Anabina and nostoc are cyanobacteria. Cyanobacteria is a type of archaebacteria or normal bacteria.
Normal bacteria.
Yeah. Hello bacteria.
Normal bacteria.
No, halobacteria means they are living in high saltic environments. Then halogens,
Archaebacteria.
Archaebacteria. Okay. Pseudomonas is a normal bacteria. So we learn these names in unit 3. Pseudomonas. pseudo. Right? Pseudo not. It should not pseudo. Pseudomonas. Pseudomonas is a normal bacteria. Then we can ignore 3 and 5 answers. Ne? 3 and 5 we can ignore. Ne? We need prokaryotes. The second one, histone proteins bound to DNA means prokaryotes or eukaryotes.
Eukaryotes.
Yeah. Then amoeba.
Eukaryote.
Protist. Halobacteria.
Bacteria.
Halobacteria is an archaebacteria. Prokaryote, therefore incorrect. Second one can remove. Salmonella is a bacteria, normal bacteria. Cycas is a tree. Eukaryote. Anabaena is a bacteria. It is a cyanobacteria. One also can be removed. Fourth and first. Several forms of RNA polymerase. Several forms of RNA polymerase. It is not included in your chart. Several forms of RNA polymerase. That means archaea or eukarya. Archaea or eukarya. Answer is number 1, Methanococcus. Several forms of RNA polymerase. You will learn in it. Learn it in Unit 3. Answer is number 1. First, structured essay question. Can you explain? I'm waiting for you. Yeah.
Following are some characteristic features of living things. State what each characteristic mean.
Mm.
Growth, irreversible increase in dry mass.
Irreversible increasement of dry mass of an organism is called as growth. Development.
Irreversible changes that occur during the lifespan of an organism.
Irreversible changes
that occur during the lifespan of an organism.
Okay. Lifespan of organism is the development. Reproduction.
Ability to produce offspring.
Yeah, ability to produce offspring. There are four main groups of organic compounds found in living things. Mention the main group of organic compounds most commonly found in each of the following. That means you have to mention each major organic components of compound of 4.
Coconut oil lipids.
Coconut oil lipids. Yes.
Egg white proteins.
Yeah. Mention.
Albumin.
Albumin. Primary cell walls.
Carbohydrate.
Yeah, very good. Carbohydrates. Mention. Primary cell wall of plants. Can you mention? What are the carbohydrates? Primary cell walls of plants. Can't you hear me up to now?
Can hear.
Aha, okay. Cellulose, hemicellulose, pectin, like that. Then, no, sometimes you can't hear me. That is why I ask. Then, what about the cell wall material of fungi? You know it.
Carbohydrate.
Yeah, chitin. Mention one laboratory test to identify the following.
Major Major organic compound group in egg white, biorate test,
Yeah. What is the major organic compound?
protein,
Protein. Major group of organic compounds found in coconut milk. Coconut milk.
lipid,
Again,
lipid,
lipids. Which kind of test can be used?
Sudan 3 test,
So then 3 test. Main storage food of plants.
starch test,
Starch test can't be right. We can't write it as starch test. How we can identify starch? Which kind of test we have to use?
iodine test,
Yeah, you have to write it as iodine test. Which type of observation can be seen in iodine test?
dark purple color,
Yeah, dark blue or purple coloration. The cell wall of fungi.
carbohydrate, chitin.
Chitin. Actually,
Teacher, that is the previous question.
reducing
Yeah,
sugars. Reducing sugars. Test name.
Benedict's test,
Benedict's test. Name 4 monosaccharides according to the number of carbon atoms and give an example for each.
3 carbon triose glyceraldehyde, 4 carbon tetrose erythrose, 5
Glyceraldehyde.
carbon
Glyceraldehyde and other one is?
triacyl glycerol.
No. Triacyl glycerol means triacyl glycerol. Glycerols. Lipids. Glyceraldehyde and the derivative of glyceraldehyde.
Phosphoglycerol.
Yeah. Glyceraldehyde and phosphoglyceraldehyde. Triose, tetrose.
Erythro.
Continue.
5 carbon pentose ribose.
Ribose. Yeah. Other examples. No need to write.
Ribulose. RUBP.
RUBP means?
Derivative of ribulose.
Yeah. How we can name it? Ribulose. RUBP means?
Ribulose bisphosphate.
Yeah. Very good. Ribulose bisphosphate. And deoxyribose as well. 6
carbon hexose glucose fructose galactose.
What are disaccharides?
Sugars formed by joining two monosaccharides by a glycosidic bond.
Glycosidic bond. Removing one water molecule is not needed or need. In your definition, it is not included. The resource book. In your note, theory note.
Notice.
No need. Then can you mention the which kind of molecules need to hydrolyze disaccharides to monosaccharides?
Catalyze enzymes.
Yeah, catalyzing enzymes or
Lysine
enzymes. State the characteristics to common all monosaccharides and some disaccharides.
Reducing sugar.
Yeah, we have to predict the third answer according to the fourth question. Describe a simple laboratory test used to identify sugars with the characteristic stated in the answer to the above question. After doing this, we have to predict the specific characteristics.
Then can't we write water soluble crystalline those characteristics also?
Yeah, but they are correct. But in some exams, they have actually here I have given the fourth question. Here it says describe a simple laboratory test used to identify sugars with the characteristic. It should not characteristics. characteristic stated in the answer to the above question. Then we can't explain any laboratory test to explain crystalline nature or water solubility like that. That is why. Then
Okay, teacher.
definitely answer should reduce in sugars. In some exams, they may give negative marks for writing another answers than the expected answer. Therefore, be careful. Describe a simple laboratory test used to identify sugars with the characteristic stated in the answer to the above question. That means you have to initially you have to write the test name, what are the ingredients, what are the regions, what about the process steps, and what about the color observation. What about the observation? Then how you can write it?
Take 2 ml of sugar solution and pour it
Yeah,
to clean
initially,
test.
yeah, very good. First of all, you have to mention the laboratory test name. Test name is what? 1 mark or 2 mark or like that.
By rate test. Benedict test.
Benedict test. Benedict test. Which kind of solution we need to do the Benedict test?
Benedict solution.
Benedict solution, also known as
Valine solution.
if you want, you can mention the color of the Benedict or failing solution due to the presence of which kind of ions.
Okay.
Benedict or failing solution is which color? In color.
Blue color.
Which blue? Dark or dark like starch amylase complex, like dark or light?
Light.
Light, due to the presence of copper 2 plus ions. Then what are you doing today? Yes, then
Mention the steps.
Mm.
Take 2 ml of sugar solution and pour to clean test.
No worries. I can understand you. No need to make your mic. Then
Add 2 ml of Benedict's reagent. Place the test tube containing the reagent in a water bath. Boil the sample for 2 to 3 minutes. When heated, positive observation is observed. The sample is turned to brick-red precipitate. It can be concluded that the above sugar sample contains a reducing sugar.
it is
Reducing sugar.
reducing sugar. Okay. Yeah, that's enough. Then what about your MCQ marks?